crosses, so if there is the statistical measure, if it crosses the set threshold which has been set in the algorithm, then a signal is declared for a given drug associated. So that is how the detection happens. So the signal is detected if the threshold is crossed. Then all signals deemed worthy of investigation require further analysis using all the available data in an attempt to confirm or refute the signal. If the analysis is inconclusive, additional data may be needed, such as a post-marketing observational trial. Next slide, please. These are the common signal detection methods, which are the statistical methods like proportional reporting ratio, the reporting odds ratio, then the Bayesian confidential neutral network, which is used by the WHO, and the multi-item gamma Poisson shrinker model, which is used by the FDA and MHRA. Then there is signal of disproportionate reporting, which is also called SDR. So SDR is a complex mathematical tool which finally gives you a data based on calculating all the. So the basic of SDR is that they use that what is the background incidence of that adverse event. If a particular adverse event is already there in place, so they use the background incidence of that particular incident, adverse event, and compared with what actually was reported after using the drug. And based on that mathematical tool, there is mathematical formulas that is being used, and then that is calculated. So if there is this usually takes form in the form of specified threshold that the ratio or other statistics must exceed. But when these rules are satisfied that the reporting ratio is greater than 1, then that is called a signal of disproportionality reporting. So the idea behind this is basically that when an adverse event is caused by a medicine, it is reasonable to assume that it will be reported more often than the background incidence. And so the reporting ratio will be greater than 1. So this is just a tool which is being used for detection. So next slide, we have what actually is the. Yes, this is the table. So this is sort of table. This is not for us to go into detail, but this is the formula which is being used. And after applying this formula, we get the information if there is any disproportionality reported. Then next slide, please. So these are the examples of safety signals, that what exactly are safety signals. So these are a few examples that any new serious unexpected adverse event or any increase in frequency of an expected event, occurrence of adverse events thought to be extremely rare in the general population is being seen. If there is any new drug or drug device, drug food or drug food supplement interactions, so any new drug with interaction with food or device. Next slide. This is a continuation. Any identification of a previously unrecognized at-risk population, the special population like children, elderly, or a different ethnic group, etc. And confusion about a product name, confusion, package labeling. So these are all come under product quality or the manufacturing defects. Even that can qualify as a signal. And concerns arising from a way a product is used, that is could be the medication error, the device use, the off-label use. So any concerns from the way a product is used, or any other concern which is identified by the sponsor or a regulatory authority. So this is not a complete list, it's endless. So these are just common examples of what a safety signal can be. So next slide, please. This table is just for your reference of what we discussed till now. That how do we see, like we were when we were talking about the SDR also. So this is what we were talking, that suppose the event incidence in the patients who are taking the product is common, and the background incidence we know is rare. For example, this was fucomelia due to thalidomide. Then the ease of proving an association becomes very easy, because all you have to do is that we can go back and have the ICSR review, and that can be compared and seen. Then again, if both in the background and the actual incidences both are rare, even then it becomes easier. It's not very easy as the first one, but then still it is easier because both of the incidence is rare only. So for that also you can have like ICSR review of a larger database because it is rare, but then that is an easy way of doing it. If it is common, then if a drug event is common in background as well and it is common in the patients as well, it becomes difficult to determine whether it is being caused by the drug actually or not. So for this, you have a very large data is required so that a substantial result can be generated. So for this, data mining is being preferred as the detection method. Then again, if the scenario is that it is uncommon in patients, but it is common to rare in the background, then again it becomes very difficult to actually find the causal association with the drug. And again, for these scenario, the bigger data mining becomes the best way of detection. The last scenario where it is rare in the patients and it is common, then it is virtually impossible because, as you can see, it is a reverse scenario. So this is most likely not with the drug, and so it becomes virtually impossible to detect any such scenario. Next slide. The newly identified safety signals are something new, which is by the US FDA Center for Drug Evaluation. They issued a new manual of policies explaining the internal process for identifying, evaluating, and resolving the newly identified safety signals. According to FDA, it is meant to provide a high level overview of how and when communication related to a newly identified safety issue is transmitted between different offices and discipline. So it applies to safety signals related to the approved drug and biologics, marketed yet unapproved drugs, over the counter monograph products, compounded drugs, and medicinal cases. FDA, as per FDA, NISS, they are serious adverse events, medication errors, or adverse events that suggest therapeutic inequivalence or quality issues that warrant further investigation. It also includes quality issue that could negatively affect public health or benefit risk profile of a product and cannot be resolved through existing routine processes. So they lay out a three-phase process for evaluating and managing NISS and explains responsibility during the each phase. They also set time frame for evaluating NISS based on the level of risk identified, which is 6 months for important potential risks and 12 months for potential risks. If it is characterized as an emergency, then the evaluation time frame differs. The next slide, please. So during the pre-evaluation phase, the signal identifier is tasked with determining whether the potential safety signal is a newly identified safety signal. So basically, it is the triaging part. That is the first phase, the pre-evaluation phase, in which it is being triaged that if it is, this could be matched, not exactly, but this is kind of what we say that after detection, the signal has to be validated. So it is somewhat the same that we have to triage the NISS for further evaluation to determine whether actually it was a safety signal or not. Then the second phase is the evaluation phase, where a safety lead is named to manage the evaluation and made the recommendation to the signatory authority that will ultimately make a final decision on the NISS. And during the evaluation phase, the team will determine the risk of safety signal, whether regulatory or compliance actions are necessary, and whether a drug safety communication is warranted. So, lastly, when it moves to the action phase, the signatory authority is taken with the coordinating the implementation of any actions which was identified during the evaluation. So, once all these three phases are completed, the outcome is documented and the NISS is closed. So these are the three phases for the newly identified safety signal. This is outlined by the FDA. Okay. So after identification, once the signal is validated, then after validation, it moves to the evaluation phase. So once they are identified and validated, they need to be evaluated further to identify the safety impact they pose on the drugs and to take any further actions, whether uh, needed. So, signals need to be evaluated to distinguish between a true and a false signal. A true signal is the real safety concern which is posed by the use of a drug, whereas false signal may be generated by the progression of underlying disease or any other medication the patient was taking. Next slide, please. Coming to the strategy of signal evaluation. So, mostly signal evaluation comprises of looking into the ICSRs for that particular event in drug and looking at the literature other than the statistical tools. Like the qualitatively, these are the ways of evaluating it. So, evaluating signals involves looking at the ICSRs and assessing the strength of causation of drug in each case. The usual sources of ICSR are unsolicited sources, which is the spontaneous reporting. The solicited cases are any organized collection of data, the contractual agreements between the intercompany exchange of safety data, and the regulatory authority sources, any ICSR originating from the regulatory authority submitted to a company. Other sources of safety data are also usually checked at this point. For example, the data from the clinical trials and any toxicology studies being done. So, when we evaluate a signal, excuse me. During evaluation, what are the points which should be considered? Is that the first thing, that previous awareness? Did we have any idea about this particular adverse event? Was it being evaluated before? But this is the first time this is being evaluated. So this is the first thing that needs to be searched. That is there any existing data? Is there any previous awareness of that particular event? Then the second part after that comes the strength of evidence. So the strength of evidence is being determined by obviously what we discussed, that we'll be looking at all the ICSRs individually for the strength of evidence. Other than that, we have to look at the pharmacological, biological plausibility, the mechanism of action of the drug. Do you think that based on the mechanism of action of the drug or whatever actions is being caused by the drug, it can probably lead to this? So the strength of evidence is based mainly on these things. Do you have any literature which do agree with the what we are investigating? So these things, these points provide the strength of evidence for that particular topic. Then clinical relevance and context. So clinically, how relevant that event is and how context, like based on the disease population, based on what population the drug is being received. So what is the relevance clinically of that particular event? Then additional sources of information may provide further evidence for or against a causal association or a new aspect of a known association and may be considered during further assessment of the signal, depending on their relevance for the signal and availability or to each organization. These may include clinical trial data, findings regarding similar cases in the scientific literature, which we just discussed, including information on substances of same class of medicinal products. So, as we were talking about strength of evidence or the clinical relevance, so the class of drugs also become important. If the drug is belonging to a particular class in which the other, suppose it is antibiotic group of macrolides. So, if erythromycin was having a reported a particular set of adverse events, and now it is being reported in azithromycin, then there must be some common mechanism of action being in the same group drug class that is plausible. So this way we do assess for the relevance and strength. So, same class of the medicinal products, information on the epidemiology of the adverse reaction or underlying disease, the experimental and or the non-clinical findings, and then database with larger data sets will always give a better idea. The healthcare databases that may provide information on characteristic of exposed patients and medicines utilization patterns. So if there are healthcare databases, they always have better information and complete information for a medical assessment. Then information from other regulatory authorities worldwide. Next slide, please. So coming to the main, which we were discussing, that ICSR review of the event always stays the mainstay of evaluation. So these are some of the type of reaction which we usually see. One is the type A reaction, which is augmented, which can be predicted from the known pharmacology of the drug, and these reactions are dose dependent. For example, bleeding with anticoagulants. Then there could be type B bizarre reactions, which are not predicted from the pharmacology of drug. They appear but are actually not dose dependent, and very small doses might also elicit symptoms. They include immune mediated side effects like maculopapular exanthema, but hypersensitivity reaction, aspirin induced asthma being an example. Then type C are chemical or chronic type of reactions which are related to the chemical structure and its metabolism. For example, paracetamol hepatotoxicity. Type D are delayed reactions which appear after many years of treatment, most common example being carcinomas. And type E are withdrawal, which occur after drug withdrawal, for example, seizures after stopping phenytoin. So these are the type of reaction, the type of events that could be reported. Next slide, please. So before starting the evaluation, there should be a signal prioritization done as well. Suppose if you have a lot of signals coming, so the signal should be kind of prioritized. So this is not triaging. There is a difference between signal triage and signal prioritization. So signal triage, if we are saying that is before validation, that is the initial when signal is detected just to see whether it is an actual signal or not, even like before validation. Signal prioritization is for validated signals, that when you have a lot of signals, many signals, so we prioritize the signals based for the further evaluation based on like the severity, seriousness, outcome, and reversibility of the adverse reaction and the potential for prevention, the patient exposure, and the estimated frequency of the adverse reaction. If the exposure is in vulnerable population or population with different patterns of use where appropriate, and the consequences of treatment discontinuation on the disease under treatment or availability of different therapeutic options, the expected extent of the regulatory interventions, for example, addition of adverse event or warnings, contraindication, etc., in the label, or whether the signal is likely to apply to other substances of the same class of medicinal products. And in some circumstances, that could cause media attention or public concern. For example, event after a mass immunization program may deserve a special attention. So these are basically to manage the time frame for further management of the signal, and it will depend on the prioritization. So any appropriate measure should be considered at any stage if information available suggests that there could be a risk that requires prevention or minimization in a timely manner. So prioritization, as we said, that if there are multiple and there are time constraints, so basically to see which may deserve special attention and time frame should be short for that management. So signal prioritization is being done based on these points. Next slide, please. So after the signal is evaluated, it is either confirmed or refuted. And there are also PRAC is the risk assessment committee. So there are certain recommendations on signals from the PRAC. If it is coming from the health authority, if signals are coming from the health authority, it is by default validated. And there are certain recommendations which we get from if it is coming from the PRAC. So these are the examples of the recommendations which are being asked from the sponsor, that the marketing authorization holder, they either ask to provide additional data for assessment within the signal procedure, or provide a review of additional data in the next PSUR, or maybe submit an ad hoc PSUR. The MH should update the product labeling information, or should request to submit a RMP, or if an existing RMP is there, to update the RMP. The MH can be asked to implement additional risk minimization measures such as educational materials or dissemination of a DHCP letter. The MH can be asked to even conduct a post authorization study according to the agreed protocol and submit the final results. There could also be some recommendations for the member states. For example, they can ask for further additional analysis, or the EMA expert group should be consulted, like the further scientific committees or expert groups to be consulted. Even an inspection can be asked from the PRAC. So these are just few examples which it can be requested from PRAC to the MH. And sometimes if there is nothing, then even we can get the recommendation of that no action is required other than the routine pharmacovigilance activities. So in the next slide, we have an example of one particular product. So this is how the PRAC uh, recommendation looks like. This was for the drug sunitinib, and the signal was for cholecystitis. So the PRAC requested a labeling change in sections 4.4 and 4.8 of the SMPC to update. So the recommendation was that MAH should perform a cumulative review of cases of cholecystitis and related terms associated with sunitinib, with particular focus on A calculus and emphysematous cholecystitis, and review should be submitted within 2 months of these conclusions. So in case the MH considers the need to update the product information further to this cumulative review, please be reminded of your responsibility to submit a variation under this. So as you see, this is how a signal request from a authority comes. Like the signal is being generated, which is cholecystitis, then you have a time frame of 2 months to perform the cumulative review of all the cases. In this, usually literature review is also done, but since here only cases are mentioned, so this. And after conclusion of these all cases, a cumulative review of all cases, then whether a labeling change of these particular sections 4.8 or 4.4 is required or not, the rationale is being presented by the sponsor. So this is how it looks like. Next slide, please. So summing it up, our presentation signal has these 5 steps. First is signal detection, then signal is being validated, then signal analysis and prioritization, then signal evaluation or assessment, then any recommendation for action, and then exchange of information. These are the signal detection that could be qualitative, quantitative. The Bradford Hill criteria is for assessing the cases in which these points are seen: the strength, consistency, specificity, temporal association, biological plausibility, coherence, reversibility. So this basically sums up the evaluation. And in the next slide, we have a flow chart, again, summing up the things that first a signal is validated. If no, then it is considered a non-validated signal. If yes, then further assessment considering all the available data. After evaluation, if there is any new or changed risk. If no, then it is a refuted signal. If yes, then proposes action such as change of the product information or any other risk minimization measures or any other whatever is being based on the evaluation. Follow up, post signal detection. Let's set up a study specifically. These are usual, this is not dictum. that sometimes even it is. So these are a few things which is done after segmented post follow up, that there could be a study specifically investigating that particular safety concern, or review of the benefit or risk of the product, to suspend or revoke the marketing authorization if it is something very serious, to propose a change in the product information, the product label, to issue a warning to the health professional in the form of a DHCP letter, or perhaps just keep the issue under review under the routine PV activities. The subsequent slides are just for reference.